I work in a project where we want to understand how genes are regulated in skeletal muscle development.
We have research problems focusing on mostly diseases, like cancer, etc. And we are trying to find new diagnostics or drugs for these diseases using computational methods. And for the support part, we maintain some of the compute infrastructure, we maintain software that's related to scientific research, and also help people through trainings or mentorships.
The main research focus of our group is to elucidate signaling mechanisms that underlie developmental processes during the development of the heart, the brain, and the entire head structures.
We're focusing on oxygen sensing of the cell and also the reaction to the lack of oxygen.
My group is interested in understanding mechanism and therapeutic approach for cardiovascular disease and cardiometabolic disease, in particular in heart failure and in obesity-related cardiometabolic conditions.
Our group works on autophagy in the immune system, and autophagy is one of the major degradation processes in the cell.
Our lab focuses on understanding the genomic instability in neuroblastoma, that is an aggressive pediatric cancer.
I'm working on how the body recycles dying and dead cells. We lose about 10 billion cells a day to cell death, and the body needs a really efficient recycling system to clear those out.
It's our job to catalyze image-based science within the Helmholtz community. That means that we support scientists to process their images in a streamlined way and make sure that the results are validated properly.
The ideal outcome of our work is to find new, more efficient, cheaper diagnostics or drugs for diseases like cancer or heart disease, which is the most high mortality diseases around the world.
We aim to pave the future until personalized medicines, since one of the aims is to include long-read sequencing in our project, and that will give us a map of patients. And by including novel approach of nanopore sequencing, we are doing like an affordable and on-time way of giving diagnosis.
Oxygen sensing is likely to be involved in several diseases. Therefore, I hope discovering a novel oxygen sensing pathway will pave the way to discover novel drugs in, for example, cancer research or vascular diseases.
Our group wants to understand the process of autophagy better to help people to improve the immune system, for example, during vaccination.
We are the link between someone's scientific idea and their practical application based on image data. This can help your doctor to distinguish faster whether your cells are healthy or sick based on their visual appearance.
When the recycling process of dead and dying cells breaks down, this can lead to several quite serious diseases, for example, cardiovascular disease or neurodegenerative disease. And so we would like to understand the mechanisms that underpin how this functions normally and how it functions when things go wrong, so that ultimately we can help to treat those diseases.
We investigate which genes are responsible for birth defects, and also how the genes together with environmental aspects play together to lead to these defects in newborn children.
My project is a part of a big collaborative effort where we want to find the most important regulatory regions in skeletal muscle development, where ultimately we then can look in hotspots of mutations of patients with congenital muscle diseases.
Simply put, we ask question how we can modify metabolism in cardiometabolic disease to impact on the syndrome. And we develop translational approaches finalized to lifestyle intervention, prevention, and therapeutics to tackle this problem.